everyone, welcome back to my class. This is Romana Ali. I hope you all are doing well. In today's class, let us talk about the process of urine formation. We have already seen the process by which the urine formation takes place. So, let us recall that. We know that aorta is the biggest artery that is found in our body, and through this artery the blood is getting circulated throughout the different body parts. The aorta, it is going to be branched into different arteries, and that arteries are responsible to carry the blood into the different organs of a human body. So, here in the kidneys, nephrons are the structural and functional units that are present. So, in the nephron, the blood is getting circulated by a branch of aorta called efferent arteriole. So, this efferent arteriole carries the blood, and that blood reaches into the structure called glomerulus. Once the blood reaches the glomerulus, the different substances that are present in the blood, which are waste like urea and water, it is going to be absorbed by the Bowman's capsule of a nephron. Once it is absorbed, then the filtrate which is present in the Bowman's capsule, it is reabsorbed by the blood, because whatever the filtrate we get in the Bowman's capsule, that not only contains excretory products, but it also has some useful products like glucose. So, it needs to be absorbed back by the blood. So, that process happens, and we call this process as tibular reabsorption. Once the tibular reabsorption is done, now the blood which is reabsorbed by these peritubular capillaries, it contains some waste products in it, but this waste products has to be transported into these proximal convoluted tubule or the distal convoluted tubule of a nephron, right? So, these waste products are again secreted by the renal tubule of a nephron, and we call this process as tibular secretion. These are the three main steps in the process of urine formation. One is glomerular filtration, the other one is tibular reabsorption, and the third one is tibular secretion. So, this is due to the ultrafiltration that is going to happen in the glomerulus. Once the ultrafiltration occurs in the glomerulus, then the filtrate is transferred to the Bowman's capsule, and after which the tibular reabsorption and tibular secretion are the steps that happens. Now, during the process of ultrafiltration, we have already seen what are the different terms like GFR and NFP. GFR stands for glomerular filtration rate, and NFP is the net filtration pressure that we have already discussed in the last session while talking about ultrafiltration. So, this glomerular filtration rate changes based on the net filtration pressure, and when the blood pressure increases in our body, at that time will the blood pressure that is found in the nephron also increases? No, that won't happen. If the blood pressure in the body, in the general circulation, if the blood pressure is high, then it would not be high in our kidneys, because kidneys have some mechanisms by which they can control the blood pressure. They can make the blood pressure normal. So, let us see what are the different factors that affect the auto-regulation of glomerular filtration rate. So, let us see how the auto-regulation of GFR occurs in the nephron. How it happens, let us talk about. So, when the blood pressure is high, normally in the general circulation, this represents the general circulation, the other different organs of our body where the blood is getting circulated. When these organs shows that the blood pressure is high, then what is going to be happen in our kidneys? Now we will see. Firstly, we will see what is the meaning of blood pressure. Blood pressure is nothing but the pressure which is exerted by the blood on the walls of the different arteries. The blood is flowing in a high speed, that it is stretching the walls, pushing the walls of the different blood vessels. That is called as, that exerts some pressure, and we call that pressure as blood pressure. When it becomes high, at the time the net filtration pressure and glomerular filtration rate, both of these get increases in the nephron. Due to the increase in the glomerular filtration rate and net filtration pressure, the things that are going to see here is stretching of efferent arteriole would occur. If the blood pressure is high, then the blood is flowing in a high speed. At the time, more amount of blood enters into the efferent arteriole. As soon as the blood, more amount of the blood along with the pressure enters into the efferent arteriole, then the walls of these efferent arteriole stretches, it stretched and it forms a bulged like structure through which the blood would enter into the glomerulus. Now, when you see the structure of efferent arteriole, then efferent arteriole shows some walls as its outer covering. It is walled. The arteries are always walled one. So, the efferent arteriole shows the walls, and on that walls you can see the presence of some channels, and these channels are called as calcium channels. So, you can see the presence of calcium channels on the walls of efferent arteriole. Now, what happens when the blood enters with a high speed into the efferent arteriole? Firstly, the stretching of the wall, stretching of the efferent arteriole takes place. As soon as the stretching of the efferent arteriole occurs, at that time, these channels, they get open. When the blood is flowing with a normal blood pressure, at that time, these channels were closed. They don't get open. But as the pressure is exerted on the walls of the efferent arteriole, when the blood pressure is high, due to that pressure, the stretching of the efferent arteriole takes place, and due to the stretching, these calcium channels which are present in the walls of the efferent arteriole, they get open. Now, when these channels get open, the calcium that is present outside, it can easily enter into the efferent arteriole. It can easily enter into the walls of the efferent arteriole, and we should know the function of calcium. Calcium is an element that is responsible to cause the muscle contraction. So, when the calcium enters into the efferent arteriole, it results in the contraction of efferent arteriole. So, in the initial time, when the blood pressure is high, during initial hours, the efferent arteriole, it gets stretched once. Once it gets stretched, calcium channels which are present on the walls of the efferent arteriole, they get open. As soon as they get open, the calcium would enter into the efferent arteriole, and this calcium causes the contraction of efferent arteriole. So, the stretching of the efferent arteriole can be prevented by the opening of calcium channels, and when the calcium enters into the efferent arteriole, it causes the muscle contraction. So, this efferent arteriole, when it gets contracted, it comes back to its normal position, and the blood which flows through these efferent arteriole, it would also becomes normal, and the normal blood flow can be seen in the efferent arteriole. There will be no increase in the net filtration pressure, as well as there will be no increase in the glomerular filtration pressure. This is how the auto regulation of glomerular filtration rate can be seen in the nephron. Now, the blood which enters into the glomerulus, it is entering into the other arteriole, that is efferent arteriole, and efferent arteriole is always less in diameter when compared to the efferent arteriole, and the blood from the efferent arteriole enters into the peritibular capillaries. In the peritibular capillaries, when the blood enters, the oxygen which is present in the blood, it can be supplied to the renal tubule of our nephron, and then the deoxygenated blood would be collected by the renal vein, and it would transport it to the heart. This is how the circulation occurs, and this is how, when the blood pressure is high, at that time the auto regulation of GFR takes place, and the kidneys would help to maintain the blood pressure in the structural and functional unit, that is in the nephron, so that there will be no frequent urination occurs. If the blood pressure is high and if the auto regulation of GFR would not happen, then in that case the blood will flow with a high speed in the glomerulus, and this causes the increase in the glomerular filtration rate. And if the glomerular filtration rate would get increases, the rate of urination would also get increases, and the human urinates frequently. So, just to prevent the frequent urination, auto-regulation of GFR takes place in our body, and kidneys helps to carry out all these things, by which the auto-regulation of glomerular filtration rate is possible. This all thing can be seen in the case 1, that is when the blood pressure is high. When the BP is high, at that time all these things are going to happen just for the auto-regulation, that is to maintain the blood pressure in the nephron. Now in case 2, we can take the situation, that is when the blood pressure is low, then what happens? How the kidneys would help, how the kidney shows the different mechanism, how it is going to help to carry out the auto-regulation of GFR. Let us see now. Let us see how the auto-regulation of GFR takes place in case 2, that is when the blood pressure is low. So, when the blood pressure is low in the general circulation, at that time and the blood flows with a very low pressure, when it enters into the efferent arteriole, then it reaches the glomerulars, and from the glomerulars the filtrate enters into the Bowman's capsule, and that filtrate contains all the different compounds along with the salt substances, along with the salt molecules, we can say. So, that salt molecules, as the blood, as the filtrate, when it enters into the glomerulus, it reaches proximal convoluted tubule. When it enters into the proximal convoluted tubule, here some of the cells are present that are specially designed to absorb the salt particles. So, here when the filtrate enters into the proximal convoluted tubule, that filtrate is going into the proximal convoluted tubule with very low pressure. So, as the blood is flowing with very low pressure, the peritubular capillaries would be able to reabsorb all the salt molecules easily, which are found in the filtrate. So, the salt molecules can be reabsorbed by the peritubular capillaries in an easy manner, as the blood pressure is very low. When the filtrate enters into the loop of Henle, then it is transferred into the DCT, that is distal convoluted tubule. In the distal convoluted tubule, some specialized cells are present. We call these cells as macula densa, and they are salt lovers. So, they need salt molecules, but when the filtrate reaches these macula densa, at that time the filtrate doesn't contain any salty substances, because the salt is already absorbed. It is already reabsorbed by the peritubular capillaries. So, it won't be available in the filtrate. When the filtrate reaches into the macula densa, when the filtrate reaches the macula densa of DCT, it won't find the presence of salt molecules in the filtrate. Now, as these macula densa are salt lovers, they need salt. Now, if they do not get any salt, if they do not get any salty molecules in the filtrate, they start informing, they start sending the signals to the efferent arteriole. So, in the walls of the efferent arteriole, some more specialized cells are present, and we call these cells as Juxtaglomerular cells. Juxta means near. So, the cells which are present near the glomerulus, that cells are called as juxtaglomerular cells. When the signals reaches these juxtaglomerular cells sent by the macula densa, then these juxtaglomerular cells are responsible to release certain chemicals in order to maintain the blood pressure, in order to do the auto-regulation of GFR. As the GFR gets decreases when the blood pressure is low, now it needs to be auto-regulated, right? So, for the process auto-regulation of GFR, these juxtaglomerular cells are going to release a chemical, and we call that chemical as renin. So, here the diagram is given. This is our efferent arteriole. and here the juxtaglomerular JG cells are present. This is our macula densa, which is found in the DCT, distal convoluted tubule. This macula densa sends signals to the JG cells. Once the signals are received by the JG cells, they release a chemical called renin, and here renin is with single N. I am not explaining about the digestive enzyme, that is RENNIN. That is another one which is found in our stomach. Renin with double N is the enzyme which is found in the stomach, and that helps in the process of digestion. But here renin with single N is another chemical. It's a different chemical that is released by the JG cells, juxtaglomerular cells, and this renin helps in the auto-regulation of GFR. Now, once the renin is released, there are so many reactions that can be seen. Enzyme is released by our liver, that is angiotensinogen, which is present in an inactive state. This chemical is present in an inactive state. Renin goes to this enzyme, renin goes to this chemical, and it starts breaking down. So, once the breakdown of the angiotensinogen takes place in the presence of renin, it is going to form a product called angiotensin 1. Angiotensin 1 is a substance which is not highly active one. So, this angiotensin 1 again, the breakdown of the angiotensin 1 again occurs, that too in the lungs. In the lungs it is going to happen. This angiotensin 1 is breakdown in the presence of another enzyme, that is angiotensin converter enzyme. It is functioning like a renin for the breakdown reaction. So, angiotensin 1 is broken down by the enzyme angiotensin converter enzyme, and it forms angiotensin 2. Now, this angiotensin 2 is an active substance that functions actively to regulate the GFR. That is, in the process called auto-regulation of GFR, it is going to help. And in the next session, I will explain you the function of angiotensin 2. Let's have a quick recap about case 2. When the BP is low in general circulation, at the time blood enters into the efferent arteriole with low pressure, NFP and GFR, both of them will be low in the situation. Then the salt molecules which are present in the filtrate, it is absorbed, it is reabsorbed by the peritubular capillaries, and when that filtrate reaches the macula densa cells, these macula densa cells are the salt lovers. They won't absorb, they won't like that filtrate which is present without salty molecules. So, they send some messages to the juxtaglomerular cells which are found in the efferent arteriole. Once the macula densa send the signals to the JG cells, they receive the signals and they release a hormone called renin, and this renin breaks the chemical substance which is present in an inactive state, that is angiotensinogen, into angiotensin 1. This angiotensin 1 becomes angiotensin 2 by the enzyme ACE that is going to happen in our lungs. So, in this auto-regulation of GFR, when the blood pressure is low, this involves the role of liver as well as lungs. And we will see the function of angiotensin 2 in our next session. That's all for today. Meet you in next class. Thank you.